Good morning everyone. So today for the journal club discussion I have chosen the topic budding epithelial morphogenesis driven by cell matrix versus cell-cell adhesion. This topic was published in the journal Cell in July 8, 2021 with an impact factor of 45.5. So we know how the buds are formed through epithelial proliferation. In this, the researchers wanted to compare the two parameters. Either the budding happens through cell matrix interaction or through cell to cell interaction. So they developed the mini organoids and compared which will be helping more in budding epithelial morphogenesis. So coming to the terminologies which are essential for discussing before starting this journal club. So coming to the first one, focal adhesion kinase. It is a protein that helps the cells stick to their surroundings, that is the extracellular matrix, and plays a role in the signaling pathways for cell movement and survival as a whole. The MDCK cell lines, these are the Mandan-Darby canine kidney cell lines. These are commonly used in the laboratory experiments to study epithelial growth and proliferation, morphogenesis, and how polarity develops between the cell. Traction force microscopy, it measures how the forces are exerted by the cells, whether between cell to matrix or cell to cell, and how these forces are developed in the surrounding tissues. YAP tag signaling, these are two types of proteins which are developed through a pathway, and these help the cells proteins to move the nucleus in response to the mechanical cues affecting gene expression and the growth of the cells. And micro-patterning technique, they have developed two mini organoid models. The second one, micro-patterning, a technique to grow cells on the surface with defined shapes and sizes to study how physical cues affect their behavior. So coming to the novelty aims and objectives, this was the first study to clearly separate and distinguish how the cell to matrix adhesion and cell to cell adhesion occurred, which drive the budding of the epithelium in the salivary gland organoids. They used 3D experimental models, developed two types of mini organoids to mimic how the real organs develop and how the budding morphogenesis was done. They discovered the two pathways, as I already discussed, the cell to ECM communication and the cell to cell adhesion. And the aims of the study was to find how epithelial bud cells form. The epithelial bud cells, as we commonly say, the small outgrowths that are found on the epithelium tissue during organ development, to understand which type of adhesion was better in this role, in this process, and to explore how the shape and the force balance was established by using the traction force microscopy. Coming to the objectives, they created the organoids using MDCK cell lines. They manipulated the cell to matrix and cell to cell adhesion techniques, and they used the immunofluorescent microscopy and the traction force microscopy to study how the cells move, how they exerted force on each other and to the extracellular matrix, and compared the two conditions. When the cell is moving and sticking to the ECM and when it is sticking to each other, they compared these two conditions. And they also blocked the pathways of the FAK, YAP and tags. If these are blocked, how the budding morphogenesis will be stopped, they try to evaluate that. Coming to the materials and methodologies used, they use the Mandan-Darby canine kidney cell lines. These are the standard epithelial cell lines used in vitro. They developed two experimental collagen model, 3D collagen gel model. In this, the researchers embedded the cells in the jelly-like matrix of the collagen, and in the second one, micro-patterning techniques. Cells were placed on specified shaped, small, printed surfaces, and they were seen to see how they are growing and how they are exerting force on the cells surrounding them. And this in turn helped them to study the role of the shape and the position of the budding occurring in these mini organoids. As I already told, they tested the cell matrix adhesion by blocking or stimulating the integrin beta 1 protein that helps stick cells to the ECM, and to test the cell to cell adhesion they modified the E-cadherin protein that helps cell to stick to each other. And they used immunofluorescent microscopy to visualize these cells, and they used live cell imaging, how the cells were moving and how they change shape in real time. They used traction force microscopy to show the forces which were measured by the cells on each other, and they used the Western blot process and the inhibitors. They blocked these pathways and they identified when the budding stopped. So they did the data analysis for determining the bud size, number of buds that were formed, how the shell shape varied, and the force direction in which the cells exhibited the force. And they did the statistical tests. What statistical tests they used? Where first one was the unpaired two-stage student t-test for the control group and the integral block group for the ECM one, and for the matrix addition group and the control group for the cell-to-cell addition group. They compared statistical analysis using two-tailed student t-test. They also did the one-way ANOVA with Tukey's post-hoc test to compare more than two groups for the different levels of the drug treatment if any drug is incorporated, and for multiple time points analysis in budding development. Mann-Whitney test was used for comparing the medians, and what were the main outcomes? The results were statistically significant. Blocking of the MAP, E-cadherin and others, the TAC pathway, they reduced the blood formation. FAK and YAP activation was significant, higher during the budding stage, and when it was blocked, the budding stage stopped. And force measurements were done using traction force microscopy. So coming to the results, this study demonstrated that epithelial budding and morphogenesis and cell proliferation can occur via two distinct mechanisms. Tissues are smart enough, either they will align with the cell-to-cell adhesion, either they will adhere to each other and form the bud, or they can produce the pressure on the extracellular matrix and they will evolve the bud. So each pathway engages different force generating techniques. Both are equally important. It is not like one is better over the other. They have two different techniques that they compare. Coming to the strengths of the study, it was an innovative comparison. They used 3D realistic models for developing the two models, 3D collagen gels and the micro patterning technique. They had clear visualization techniques for using the immunofluorescent microscopy and the force traction methods for measuring the force. They had the mechanistic insights, how the different molecular pathways develop and how they induce the budding was observed. And they did the functional mechanism by the gene knockdown process and how the budding stopped. And it had broad application. We can study the organ development, cancer progression and metastasis, and we can implement it on the future studies for tissue engineering applications as well. So coming to the limitations, it is only limited to this one cell line. For it was done in the human submandibular mouse salivary gland model, so they can use the HCG also in further studies. They used in vitro conditions, so in vivo conditions should be also done after the clinical trial. Short-term observation, it was only for a limited time period. Also, they had the limited cell types and the signaling pathways, and no in vivo validation was done. So in further studies, they can do the in vivo validation. Coming to the study design, what type of study design? It was a basic experimental laboratory research design, which was exploratory in nature, mechanistic, focusing on the mechanisms, quantitative with measurable outcomes. So all the checklist items, clear research question, objectives, hypothesis implementation, experimental model description, control groups, all these were done correctly. But the blinding and the effect size measurement, reproducibility measures and the limitations acknowledged were not done in the study. So we could do the clear information could be done on the blinding, confidence intervals, data code availability, and explicit limitation discussion in the discussion section was not done. So coming to the future scope, they can test in vivo and they can explore the other cell types apart from the cell type that was being used in this. They can do long-term morphogenic study. It was a short-term study only, so long-term can be done. Investigate disease prevalence, specifically for cancer progression, tissue repair and bioengineering mechanisms. Integrate the signaling pathways such as the WNT pathway, sonic hedgehog pathway, notch pathway. They can interact, and how these help in the adhesion mechanisms and play a role they can do in the further study. Applying the tissue engineering in the further studies for creating artificial organ design and regenerative medicine responses, and develop predictive models using computational methods and the stimulation studies. We can predict the tissue behavior based on the force fraction.